Have you ever pondered what the future holds for high-performance liquid chromatography, or HPLC as it's commonly known? This powerful tool, central to analytical chemistry, is widely utilized in diverse fields such as pharmaceuticals, forensics, and environmental science, to name just a few. Its ability to separate, identify, and quantify components within a mixture has been instrumental in scientific advancements. Yet, as we stand on the cusp of a new era, we find ourselves asking, what's next for HPLC? How will it adapt, innovate, and grow to meet the evolving demands of our time? Will we see an increase in efficiency, accuracy, or perhaps even a revolution in its fundamental principles? These questions are not just idle musings. They are pivotal to the future trajectory of analytical chemistry and its allied fields. So, stay tuned as we delve deeper into the fascinating world of HPLC and its future prospects. To appreciate the future of HPLC, a clear understanding of its principles and functioning is crucial. Now, HPLC, or high-performance liquid chromatography, is a versatile and highly sensitive technique used primarily in analytical chemistry for separating, identifying, and quantifying components within a mixture. Central to HPLC are its four main components. First, we have the pump, which propels the mobile phase and sample through the system. The injector, our second component, introduces the sample into the mobile phase stream. Then, there's the column, the heart of the system, where the actual separation of compounds occurs. And finally, the detector, which identifies and quantifies the separated components. Now let's talk about the separation process. This is where the mobile phase and stationary phase come into play. The mobile phase is a solvent or mixture of solvents pumped through the column. The stationary phase is the material inside the column which interacts with the sample components. The differing interactions between the sample compounds and these two phases lead to separation of compounds as they pass through the column. Here's a simple way to visualize it. Imagine a group of people running a race, but they're running through a thick forest instead of an open track. The fastest runner isn't necessarily the one who gets to the finish line first. It also depends on how they navigate the trees, or in our case, how the compounds interact with the mobile and stationary phases. And why do we need this separation? Well, it allows for detailed analysis of complex mixtures, which can be crucial in a multitude of fields like pharmaceuticals, forensics, environmental monitoring, and even food and beverage industries. With this understanding, the potential advancements in HPLC become even more intriguing. By manipulating the mobile and stationary phases or the properties of the column, we can fine-tune the separation process, leading to more accurate and efficient results. But we'll delve deeper into that in the next scene. No technology is without its limitations, and high-performance liquid chromatography, or HPLC, is no exception. When it comes to HPLC, one of the most noticeable limitations is the analysis time. Even though HPLC is renowned for its precision and accuracy in separating compounds, this process can be time-consuming, especially when dealing with complex mixtures. The separation process involves the repeated circulation of the sample mixture through a column, and each cycle can take several minutes to hours. This long duration can be a hindrance in situations that demand quick results. Another challenge faced in the realm of HPLC is the requirement for highly skilled operators. The operation and interpretation of HPLC results necessitate a deep understanding of chemistry and the specific principles of chromatography. This means that the use of HPLC is generally restricted to well-equipped laboratories with trained personnel, limiting its accessibility for routine testing in smaller labs or field settings. The third limitation lies in the difficulty of analyzing certain compounds. While HPLC is incredibly versatile, it's not all-encompassing. Some compounds, especially those that are volatile or thermally unstable, prove difficult to analyze using HPLC. Compounds that have very similar physical and chemical properties also pose a challenge as they may not separate well on the column, leading to inaccurate results. Moreover, HPLC systems are not immune to wear and tear. Over time, the columns used in HPLC may degrade, affecting the quality of separation and potentially leading to inaccurate results. Regular maintenance and replacement of these columns are necessary, adding to the overall cost and complexity of using this technology. However, it's important to remember that these limitations are not insurmountable. With every challenge comes an opportunity for growth and improvement. The world of science is continuously evolving, and so is the field of HPLC. As we look forward to the future, these limitations serve as stepping stones, guiding us towards the development of more advanced, efficient, and accessible HPLC systems. These challenges pave the way for exciting advancements and updates in HPLC technology. As we strive to overcome these limitations, the future of high-performance liquid chromatography, or HPLC, promises exciting developments. The current trend in the world of scientific research is to go smaller and smarter, and HPLC is no exception to this. Let's dive into the future, shall we? First on the horizon, we see the advent of micro and nano HPLC. This technology aims to miniaturize the entire HPLC process. By reducing the size of the chromatography system, we'll be able to conduct more analyses in less space and with smaller sample volumes. Not to mention, the miniaturization of HPLC will bring about significant cost savings, as less solvent is required for each analysis. Furthermore, the use of smaller columns in micro and nano HPLC can increase the speed and efficiency of separations, thereby reducing the analysis time. But it's not just about size. The future also holds fascinating advancements in column technology. We're talking about the heart of the HPLC system. Scientists are currently exploring the use of new materials and designs to improve the resolution of separations. For instance, the development of monolithic columns, which have a single continuous structure, could provide a more efficient and faster separation than traditional packed columns. Moreover, the use of new stationary phase materials, such as core shell particles, could increase the performance of HPLC columns by providing more surface area for interaction with the sample. Next, we look forward to improvements in detector sensitivity. As the saying goes, you can't improve what you can't measure. Detectors play a crucial role in HPLC by detecting and quantifying the components in a sample. Future detectors for HPLC are expected to be more sensitive and precise, allowing for the detection of even trace amounts of compounds. This would open up new avenues for the use of HPLC in fields like forensic science and environmental monitoring, where detecting minute quantities of substances is often crucial. Lastly, but certainly not least, we anticipate the integration of artificial intelligence for data analysis in HPLC. Artificial intelligence, or AI, has the potential to transform data analysis in HPLC by automating the process, reducing errors, and increasing accuracy. Imagine a system that could automatically interpret HPLC data, identify patterns, and make predictions. This could significantly streamline the analysis process, saving time and resources. Moreover, AI could enable more complex analyses, uncovering insights that might not be apparent to the human eye. These advancements will revolutionize HPLC, expanding its applications and increasing its efficiency. With the integration of new technologies and innovations, we can expect to see HPLC becoming an even more powerful tool in scientific research. The future indeed looks bright for HPLC, and we can't wait to see what it holds. These advancements will revolutionize HPLC, expanding its applications and increasing its efficiency. The future of high-performance liquid chromatography, or HPLC, is not just about the technology itself, but also about its implications. Let's delve into what these advancements could mean for various fields, starting with pharmaceuticals. In the pharmaceutical world, HPLC's new capabilities could revolutionize drug discovery and development. The improved detection sensitivity and enhanced resolution will enable scientists to identify and quantify even the most complex mixtures of compounds. This could lead to the discovery of new drugs and therapies and a more efficient drug development process. Now, let's consider environmental science. The robustness and precision of future HPLC systems could greatly enhance our ability to monitor and assess environmental pollutants. With the ability to analyze even minute quantities of contaminants, we could gain a more comprehensive understanding of environmental health. This knowledge could then guide our efforts to protect and preserve our planet for future generations. In forensic science, HPLC plays a crucial role in evidence analysis. The future advancements in this technology could provide forensic scientists with more accurate and reliable data, aiding in solving crimes and ensuring justice. With the potential for faster, more efficient separations and the ability to analyze even the most challenging samples, HPLC could become an even more indispensable tool in the forensic lab. Beyond these fields, the potential for new applications of HPLC is vast. With its enhanced capabilities, HPLC could be used in novel ways to address some of the most pressing challenges of our time. From developing new materials to advancing our understanding of complex biological systems, the possibilities are endless. As we wrap up our journey into the future of HPLC, it's clear that these advancements are not just about improving a technology. They're about expanding our knowledge, pushing the boundaries of what's possible, and creating a better future. As we look forward to these exciting developments, one thing is certain: the future of HPLC is bright and full of promise.